Welcome back, life science learners, to this segment. Trust that you guys are energized and ready for this part of the lesson. We were spending some time looking at biodiversity before we went into a little break. It's important that now we look at the context of biodiversity in a new system, in a system where we're looking at the biodiversity and classifying them. Yes, in this part of the lesson, we're looking at the concept of classification. What does it mean? In life sciences, we've got systems that classify organisms. The fact that we can differentiate between plants and animals and reptiles and mammals means that there's been a system that has put individuals or organisms into specific groups. And that essentially is classification. Why is that important? Let's try and get into the lesson and unpack that. So let's look at the concept of what classification schemes are. Let's understand that due to the vast numbers of organisms that exist on Earth, scientists group organisms according to features or characteristics they have in common. And that's fundamental to any classification group. We put organisms or objects together based on specific characteristic or features. It's like basically sorting out cutlery in the kitchen or clothes in your cupboard. The fact that you at school are placed in specific classes, you know that it is a system that organizes you based on the year of your study. So you could be in grade 10, 11 and 12. That's a classification system. And within the grade 10, 11s and 12s, you've got learners that do specific combinations of subjects. So we put them into different subject modules, or we put them into specific course selections. So again, guys, we see that there's a system of classification that is used to organize. And so why do we need to keep systems in place that allow for classification? It's easier for us to be able to access individuals or information. And that is essentially what classifications have done. And so as biologists over the years have started to look at characteristics and features of individuals that are similar or different, they've created groups of or schemes of classification. And so as we see with new developments and the understanding of features and characteristics, our systems or schemes of classifications have changed over the year. Let's try and unpack all of that. So Aristotle, in between 384 and 322 BC, he was a Greek philosopher, and he had classified living things into plants and animals according to their appearance. And that was a very simple system, guys, of being able to identify animals based on features they had, and he separated them into animals and plants. I mean, it seems very basic, but he had the foresight to be able to say, well, there's a difference between animals and plants, and hence he created a group or system that allowed for the classification into plants and animals. He divided these into two main groups, as I mentioned, plants and animals. His system was used in the early 1600s. People wrote about animals and plants, either used their common names in various languages, or adopted more or less a standardized description. So guys, we know that when we talk about different areas around the world, we all have different languages. However, being able to have a common system internationally allowed for us to be able to recognize certain individuals or certain groups of individuals based on the characteristics they have. And this, back in the day, was important for us to be able to recognize that individuals that had put organisms into specific groups had used a set of characteristics, and that could be applied to anywhere else in the world, and hence created what we refer to as a classification system. Carolus Linnaeus, again in 1707, lived till about 1778. He was a Swedish scientist that introduced the binomial nomenclature system. Basically, it's called the two-name system of classification. Again, this was a system that used two names to classify organisms. Like we have a name and surname, it was a similar system that was introduced. And this was the first system that started to now bring into the concept of species and genus. Let's try and unpack that further. Again, the capital letters were used for the genus, and a small letter was used for the species. Again, we all are familiar with the species that we belong to. It's sapiens. The genus we belong to is called Homo. So Homo refers to the genus, and sapiens refers to the species. Again, this is a binomial system of classification that puts individuals into a specific genus, which is a classification group, and then into a group belonging to a species of individuals. And this is used to classify modern humans and into specific orders of systems. Panthera leo is again a binomial system for the lion, again a system made up of the genus and the species. Ernest Haeckel was born in 1834, lived up till 1919. He was able to observe under the microscope microscopic single-cell organisms, and he proposed a third kingdom of life which was called Protista. And in addition to plants and animals, he had introduced a group called Protista. It's important that we start recognizing that as technology started improving and the ability for organisms to be seen under a microscope improved, the system of classification had to be reviewed. It was then that they realized that apart from plant and animals, there were things that were microscopic that could be seen only with some magnification tool, and that opened up another dimension for classifying organisms. Protista was a single-cell organism that was neither plant nor animal, but could have had the characteristics of either. Herbert Copeland lived between 1902 and 1968, which is much more recent. He recognized the important difference between a single-cell eukaryotes and single-cell prokaryotes. So again, as the advancement of technology came through with more developed scientific tools like microscopes, these individuals were able to look at cells under a microscope and differentiate between what a prokaryotic cell was and a eukaryotic cell. He then proposed a four-kingdom classification, and he placed the bacteria and blue-green algae in the fourth kingdom called Monera. And so as we start unpacking this, we start seeing that as time changed and how the ability to be able to see structures under the microscope had improved, we started to see that groups of individuals showed a specific set of characteristics which created a separate group, and this group was called Monera. Robert Whittaker, in 1902 to 1980, more recently, devised the five-kingdom classification in 1968. He recognized that fungi belong to their own kingdom. However, even today, the five-kingdom system is still under dispute. Again, guys, as you see, as we start unpacking and we start seeing the characteristics of individuals more specifically and in more detail, we start changing our systems of classification. It is not a bad thing. It is just becoming a more refined way in which we place organisms. And that's naturally bound to happen as we start seeing the ability to identify features more stringently using more evolved and developed techniques. It is in nature of science that more discoveries will come to light and theories will continue to be improved upon and revised. And that's important that we recognize that. So guys, we've just looked at how the system of classification schemes were developed, and one thing that we've recognized is that the system of classification was never cast in stone, and we know that currently there are systems that are placed that are being reviewed constantly, and that's important for us to be able to recognize. So let's have a little break, and when we get back, we will look at some of these systems in detail, and we'll try and apply our understanding of how to use these in naming and classifying organisms. Short break, and I'll see you in a bit.